This is the second part of a two-part series. The first part focused on a background and history about the field of glycobiology and the structures of sugars. And this part, which is part two, is going to focus more on some recent research from my own laboratory at Berkeley with the goal towards imaging the glycome. And in fact, what you're looking at here is a fluorescence image of a zebrafish that's 5 days old, in which some of the sugars have been tagged with fluorescent molecules. And this allows us to literally visualize those sugars in a live organism. And so let me tell you about why we're interested in that. Okay. Now, the field of molecular imaging is certainly large and expanding rapidly, and it's very important both from the perspective of clinical medicine as well as in the research laboratory. So many of you might have found yourself lying on a table like this at one point in your lives, as have I. This is a magnetic resonance imaging scanner, or an MRI scanner, and what this scanner can do is literally take a picture of molecules inside our body without having to cut us open, which is very convenient for many of us. And there are devices very similar to this one which allow clinicians to look at other molecules in our bodies, molecules that are radio-labeled, for example, and that's what this image is here. This is called a PET scan of a human body that has been injected with a radio-labeled version of glucose. Now, these are important tools for looking at molecules in clinical settings, but we also like to look at molecules in research settings, particularly to understand what molecules are doing inside living cells. And nowadays, we have some wonderfully advanced fluorescence microscopy tools that allow us not just to see the collections of molecules in cells, but individual fluorescent molecules as well. So it's a very exciting time to be doing molecular imaging in living systems. Now, I mentioned in my first lecture that one of the most intriguing aspects of glycobiology is the fact that the glycome, or the complete collection of glycans that a cell makes, is dynamic. It's not fixed. So the glycome, or the collection of sugars on the surface of a cell when it's in one particular state, is different from the collection of sugars on that same cell when the cell has transformed its state. And this is certainly the case when embryonic stem cells choose a differentiation fate and then become a differentiated cell, like a muscle cell or a neuron. If you look at the collection of the glycans on the cell surface glycolipids and glycoproteins, you'll find that the nature of that collection has changed as the cell has undergone a differentiation process. From the perspective of clinical medicine, it's interesting to note that the glycome changes when healthy cells transform and become cancer cells. So you can understand if the sugars on the surface of the cell are changing in a manner that is characteristic of a cancerous state, it would be very convenient if we could actually see those sugars change in vivo, in living human beings, because that would offer the possibility of imaging those changes and detecting cancers when they're at hopefully very early stages. So there are many reasons why one would want to image the glycome, to take a look at how those sugars are changing inside living subjects, so we can understand how do the glycans relate to differentiation of cells or to organogenesis? And can we detect changes in the glycome that correlate with disease states so that we can detect those diseases at very early stages? The challenge, of course, is to figure out how to tag these sugar molecules with probes that we can actually visualize using imaging technologies. So even in the research laboratory, this is very challenging. The most commonly used imaging technique in research laboratories is fluorescence imaging or optical imaging. And we don't even have a way to put fluorescent tags on these sugars. So that is a challenge that students and postdoctoral fellows in my laboratory have taken on. And it's a project we started working on back in the late 1990s. So it's been progressing now for over 10 years. Just to give you a little bit of counterpoint, people who study proteins using molecular imaging techniques have a wonderful arsenal of tools available to them, which is why images like the one I showed on the previous slide are so common and so familiar to most cell biologists. In fact, most cell biologists probably spend a good portion of their day looking at images similar to this one. In this cell, 2 different proteins have been labeled with 2 different fluorescent dyes, one green and one red. And the protein that appears green has been labeled using a genetically encoded reporter. So let me just show you what that reporter is. It's called the green fluorescent protein, abbreviated GFP. And it's one of the most important tools for fluorescence imaging of proteins. The green fluorescent protein, because it's a protein, it's encoded by a gene. And it turns out that one can fuse the gene that encodes GFP to the gene that encodes the protein you're interested in imaging. And now when the GFP protein fusion is expressed, basically your protein of interest is labeled with a fluorescent molecule. The GFP has a chromophore inside that is fluorescent. And this is such an important tool. It's revolutionized our ability to study proteins, not just in live cells, but even in transgenic organisms in which we express these GFP fusions by manipulating the genome during the embryonic stem cell stage. This is so important that it was recognized with the Nobel Prize in Chemistry in the year 2008, and that prize was granted to these 3 scientists. Very well deserved, very exciting development in the field of molecular imaging. Now, those of us who study glycobiology, of course, would love it if we had a tool that was comparable to the green fluorescent protein, so that we could label our sugars with a fluorescent tag, the way that people label proteins with this fluorescent tag. But unfortunately, the biosynthetic machinery by which sugars are built doesn't really lend itself to these genetically encoded reporters. That's because sugars, unlike proteins, are not primary gene products. Each of these complex glycans is not encoded by a single gene. Rather, they are the products of a complex series of metabolic steps inside the cell. They're products of metabolism. And so for that reason, we cannot use genetically encoded reporters like GFP to introduce labels onto sugars. So I drew this cartoon as kind of some comedy because we would love to be able to put a tag like GFP on the sugars, but there's really no mechanism to do that using genetics. So in my laboratory, we've been focusing on using the metabolic pathways of the cell to introduce fluorescent probes into the sugars that are not the GFP, but rather small molecule fluorescent reporters. And let me show you schematically how we go about doing this. It's a 2-step process, and we call this metabolic labeling of glycans with chemical reporters. The first step is to feed cells a synthetic monosaccharide building block. Now, this synthetic sugar looks very much like one of the natural monosaccharide building blocks that you might find in foods that you eat. And I introduced those structures in my first lecture. But the difference is that we've altered the structure of this monosaccharide building block to introduce a chemical functional group X. And X is just the generic for now, and in a minute we'll get to what that is. But X is what we call the chemical reporter. It's a reactive functional group, so that when the cell takes this monosaccharide building block, and when the enzymes process it and integrate it into the glycans, when the glycan appears on the cell surface, that reactive functional group is now on display and can be used in a second step, which is a chemical reaction with a probe molecule. Now, the probe is a fluorescent dye, for example, and that probe has its own chemical functional group. Let's call it Y, just as a generic label. But X and Y have to be designed so they react with each other to form a bond. And once that bond is formed, now there's a fluorescent probe molecule bound to a cell surface glycan. And that probe now allows us to visualize all of the glycans that possess this red monosaccharide building block as one of their constituents. And let me tell you, there is a lot of functionality inside this cell. All of your proteins and your lipids, your nucleic acids, metabolites, there's water, there are a lot of chemical entities inside this cell. And X and Y have to be designed to avoid any side reaction with those biological groups and still react only with each other. So which sugars would you like to image? Of course, there are many interesting choices. And as I introduced in my first lecture in vertebrates, there are 9 fundamental monosaccharide building blocks that are the constituents of our various glycans. They're all interesting in their own right, but when we were thinking about what sugar would we like to image, we were naturally drawn to this monosaccharide called sialic acid. And as I mentioned in my first lecture, sialic acid tends to appear in some very interesting circumstances in vertebrate biological systems. I mentioned, for example, that sialic acid is a component of the ligand for the selectins. I also mentioned that sialic acid is the structure that the influenza virus binds to. Well, it turns out that sialic acid is a pretty busy monosaccharide because it also seems to be related to embryonic development and to cancer. In fact, the glycomes of embryonic cells differentiating tissue as well as cancer cells have been studied in much detail, and it turns out that many of the glycans found both embryonically and on cancers have sialic acid as one of their constituents. So what I've shown here are just 3 examples from a much larger set. These are glycan structures that have been found to be highly elevated on cancers compared to the normal healthy tissue counterpart. And many of them are also developmentally regulated, which is probably not an accident because we know that often cancers start to acquire properties that we normally associate with embryonic tissue. They've sort of lost their identity and they're reverting to an embryonic state. And that's reflected in their glycan structures. So polysialic acid, which is a homopolymer made up of repeating units of sialic acid, is highly abundant on a variety of tumors that are derived from neural crest tissue. Siala-Lewis X, which I mentioned before, is part of the ligand for the selectins, is also found in elevated levels on a variety of different epithelial cancers and blood cancers. And sialyl-TN, a very simple disaccharide, is actually not normally found on any healthy human tissue, but it appears on a variety of prostate cancers. And we don't really know why that is, but the fact that these structures appear in the context of malignancies has suggested to many people that there might be elevated levels of sialic acids on these tissues. So if we could image the sialic acids, maybe we could detect these tumors in living systems. That was one of the motivations to study the sialic acids. So in this experiment, what we did is we grew some cells called HeLa cells. These are human cervical epithelial cancer cells that you can grow in a test tube. And while those cells were growing, we fed them mannaz in the media. We just added it to their cell culture media. They took it up, they digested it, and they put azides into their sialic acids. Then, after a few days of treatment with mannaz, we reacted those cells with QFOS. And then after a couple of hours, we took a fluorescence image of those cells. The cells were also stained with a blue fluorescent dye that's specific for the nucleus. That just helps you to orient exactly where the cells are positioned, since each one has one nucleus. So what you can see is that each cell is surrounded by a nice bright green outline. Those are the sialic acids on the membrane-associated glycoproteins and glycolipids of the cell. And they're nicely lit up. We're actually imaging the sugars. You might see a little bit of fluorescence inside the cells as well. That's basically part of the secretory pathway, the Golgi compartment, for example. And then there's a little bit of staining of little vesicles throughout the cell as well. That's because after we react sialic acids on the membrane, some of those sialic acids end up internalized into vesicles because the membranes are constantly being engulfed and recycled and turned over. They're going back into the late part of the Golgi compartment, then coming back to the membrane. There's a lot going on, in fact, during the course of reaction with QFOS. Now, we had some issues once we started doing these kinds of experiments. For example, we realized that in order to get this nice, bright staining of the sialic acids, we had to react the cells with QFOS for several hours. Now, that's not a problem for imaging the sugars on cultured cells, because the cells are in a flask with the reagents and they can sit in an incubator for several hours and just let the reaction unfold. The reason we had to let the reaction go for several hours as opposed to several minutes or several seconds is that it turns out the Staudinger ligation reaction is intrinsically rather slow. It just takes that long for the reaction to proceed. And although that was not too problematic for imaging experiments on cultured cells, we worried that that could be a problem for imaging sugars in living animals. Because, of course, a living animal is not at equilibrium. It's not cells in a flask sitting in an incubator for many hours. Animals have an active metabolism. As soon as you introduce reagents into their bodies are working on clearing those reagents right out again. And that clearance process can be very rapid for certain molecules and certain animals. In fact, when we started looking into the Staudinger ligation as a tool for imaging sugars in laboratory mice, we immediately ran into this problem. So, what animal model would one want to use to image the sugars? Mice are obviously a very attractive model because one can study a variety of human diseases in that model organism, including cancer. However, mice are not ideal for optical imaging because, as you know, they're not transparent, they're opaque. One can do fluorescence imaging inside a mouse, but it requires a specific kind of dye, generally a near-infrared dye, and it's a more complicated experiment. And for a first attempt at in vivo optical imaging, we thought we would use a model organism that's a little friendlier to an optical microscope. And the obvious choice was the zebrafish. So zebrafish are translucent. You can see right through them and monitor their organs in vivo in the live animal. Also, zebrafish are a very attractive model for vertebrate developmental studies. They have all the same organs and parts that we have. They're vertebrates. And their embryonic developmental program has been very well characterized. So we know what zebrafish embryos should look like at virtually all stages of their development. We also know that there are changes in the glycome that correspond with embryonic development. And so we thought this was a very nice organism in which to, first of all, test our chemical tools. But second of all, we might be able to address some very interesting fundamental questions of how the glycome changes during development and learn something about the field of glycobiology. So we started with some proof of concept experiments in which we focused not on sialic acid, but rather on a sugar called N-acetyl-galactosamine, abbreviated GalNAc for short. The reason we were so interested in GalNAc is because, as I mentioned in my first lecture, it's the conserved core residue in a family of O-linked glycans that are associated with glycoproteins from the mucin family. Mucins are a general class of glycoproteins that are characterized by having dense clusters of these O-linked glycans along the polypeptide backbone. They're involved in cell adhesion, regulating cell-cell interactions, and they're known to be both developmentally regulated and sometimes upregulated during malignant transformation. So the mucins are an interesting class of glycoproteins. They all have a GalNAc residue at their core position. So we thought if we can introduce an azide into this GalNAc residue, perhaps we could image the mucins in vivo. This experiment demonstrated to us that we can in fact image those GalNAs residues in a live zebrafish. And I should point out that these zebrafish look perfectly normal, perfectly healthy and happy, despite the fact that some fraction of their GalNAc residues were replaced with GalNAs, and that some fraction of those GalNAs residues were in fact chemically reacted with a DIFO reagent. These zebrafish were then released back into their tanks, and they go on to live a normal life as far as we can see. That kind of an experiment is what led to images like this one, which I showed on the very first slide of this second lecture. So this is the head of a 5-day-old zebrafish that has been labeled with GalNAs followed by 3 different colored fluorescent dyes at 3 different time points. In this particular image, the newest glycoproteins are appearing red, and you can see there's a concentration of those glycoproteins here in the olfactory organs, which are basically the nostrils of the zebrafish, whereas older populations of glycoproteins appear either blue or green. And you can see that the blue and the green has quite a different distribution from the red. And by looking at how these different colors have moved around as a function of time, we can learn something about the dynamics of the glycome. And we think this will allow scientists to study glycobiology using all of the hardware of molecular imaging that so far has focused primarily on the study of proteins. And finally, where we are going with this technology in my laboratory is to continue our studies of developmental biology so we can understand how the glycome contributes to some of the early cell fate decision-making processes during the embryogenic program. For example, how does the glycome change when pluripotent embryonic stem cells are just starting to choose their ultimate differentiation fate? We're hoping we can capture a snapshot of that moment from the perspective of the glycome. And then finally, one of our original motivations was to develop these tools in a manner that could be clinically useful. So we're hoping that we can develop the chemical reagents, the azidosugars, in such a manner that you could actually introduce these reagents into human subjects and use them to detect tumors at early stages by virtue of their changing glycome. So stay tuned, and hopefully we'll have a lot more to say about that in the future.